Okay, thank you very much. I'd like to thank again all the organizers for hosting us here. I also want to mention that I'm going to talk a little bit about the work of several of my friends and colleagues that are mentioned in here. The talk title is a bit ambitious. I'll just give you a flavor of some of the work that we've been doing, some of the challenges that we've faced in modeling the functions of ventral visual cortex, and some of the models that we've been thinking about, and potential solutions and challenges for future work. So just to start with the basic anatomy of the visual system. So here on your right, you see one of the many examples of deep convolutional neural networks. In this particular case, for the aficionados, this is called ResNet 150. And then on the left, we have a diagram that's quite famous, and that has many decades old now, that illustrates the basic mesoscopic connectivity of the ventral visual cortex and dorsal visual cortex in the macaque monkey. And I think it's pretty clear, even just glancing at these kind of diagrams, that the sheer complexity of the anatomical connectivity is completely different, and there's clearly lots and lots of details about the architecture that are completely missing. To some extent, if we squint and we kind of don't pay attention to a lot of details, we might be able to argue that current models capture some aspects of some of the boxes that are illustrated here that comprise basic feed-forward processing along the ventral visual cortex. But even that is kind of questionable, as we've seen from some of the previous talks. So we spent a lot of time, and I won't go into details. There's a review paper that we have here trying to argue where we need to go next, going beyond the feed-forward sweep and talking about feedback computations in visual cortex and some of the challenges that current models face. We also recently conducted a Turing test survey of vision and language models, where we basically try to compare the behavior of different large-scale models, both in visual and language tasks, in terms of their output, and asking to what extent human judges, but also AI judges, are able to distinguish between answers coming from humans and machines. And this has been quite instructive to us in terms of highlighting many areas where they're quite similar, and it is indeed quite remarkable that many AI models can fool and imitate humans quite well, but also several areas where there are important differences between the two. And so for the purpose of today's talk, I'd like to focus on one example of a challenge for this family of computational models of visual system, which involves the problem of pattern completion and making inferences from partial information. So if you look at this picture here, it's relatively easy for most of you to recognize many of the objects here. And even if I look at some of these, is the mouse there? Yeah. So even if you look at some of these objects here that are heavily occluded, you can still recognize these tables and chairs quite well. In some cases, even from a handful of pixels. This, for many of you, may have not been in this particular place. Even if you have been in the famous Piazza San Marco in Venice, in Italy, you may not have been there when it was flooded like this. So this is an instance, perhaps, of zero-shot learning. You may not have seen chairs and tables that are flooded, and you can still recognize them quite well. So how is it that we can recognize objects from partial information? So we conducted a series of studies. I'm going to go through two of them in particular. In the first study, we just used very simple objects like this one, and we used a technique called bubbles to render them, to occlude them basically, and only show them show partial information. So bubbles basically amounts to looking at the world like this through small bubbles. If you have a lot of bubbles, you have a lot of information. If you have very few bubbles, you don't have a lot of, you have less information. So we can titrate the difficulty of the visual object recognition task If you do that, this is one of those examples where we did a 5-way forced choice categorization task. Chance level is 20%. The y-axis indicates performance. The x-axis indicates the visibility. If you have 100% visibility, this is a very simple task. People are at 100% correct. And here you can see that human subjects are extremely robust in visual object recognition in this family of tasks. So we can get correct recognition even with about 20% visibility. We can ignore all the different colors here, which correspond to the amount of exposure to the different pictures. And then in subsequent studies where we used a technique called backward masking, we showed that it costs about 50 milliseconds to perform pattern completion. That is, when we have these objects that are heavily occluded and you do backward masking, it takes about 50 milliseconds. It takes additional computations to perform object recognition. That's what's shown in here. So when you do backward masking, performance drops quite significantly, especially in those cases where you have very little amount of temporal exposure. With 25 millisecond exposure, people are unable to do visual pattern completion. So this cost of 50 milliseconds is also reflected at the physiological level. So here I'm showing you invasive neurophysiological recordings that some of our colleagues conducted in patients with pharmacologically resistant epilepsy, where they put electrodes inside the human brain in order to monitor activity in these patients for the purpose of treating them for epilepsy. So what you're seeing here are, in this particular case, recordings from one particular electrode in the inferior temporal cortex of one of these patients. This is the intracranial field potential on the x-axis as a function of time. These are 39 repetitions of one particular picture. You see a very strong visually selective response to this picture. When you show these heavily occluded images, you see that we can still evoke these these strong visually selective signals in the same region. But all of these signals, interestingly, are delayed also by about 50 milliseconds. So the numbers that you see on the x-axis correspond to the trough in the field potential signal. So we concluded that it costs also about 50 milliseconds to perform pattern completion at the physiological level. So we also, in the second experiment that I want to show today, we used a set of images that were introduced by Shimon Ullman and colleagues. Okay, so these images, they termed them minimally recognizable configurations, and they are extremely hard to recognize. So basically what they took is they took real object images and they started cropping them and blurring them until basically they became unrecognizable. So it turns out that you can introduce minimal changes in these images so that we can basically go from recognition to lack of recognition by just introducing either one pixel on each side or by introducing minimal amounts of blurring. So here, in the interest of time, I'm not going to do a demo, but here, what you see on the left, it should be very, very hard to recognize. The one on the right should be easier to recognize. This particular picture is part of an eye. Here's another example. This is on the left, so it should be very hard to recognize. The one on the right should be much more readily recognizable. This is a person with a tie. And these are cases where even minimal changes can make the whole difference between recognition and lack of recognition. So again, at the behavioral level, this is very robust. So these sub-Merck images, these unrecognizable images, are recognized at a rate of almost 0%, whereas the Merck counterparts, which are minimal changes with respect to the other ones, are recognized almost well above 80% correct in this case. Interestingly, in this case, after being exposed to the full object images, if we expose subjects again to the sub-Merck images, people are much better, as reflected here in the sub-Merck post condition. This is an instance again of very rapid learning by humans to recognize these images after being exposed to the whole objects. And again, at the physiological level, we also show that there are massive differences in the underlying neural representation between those images that can be recognized and the ones that cannot be recognized. So here's one example of the differences between these Merck and sub-Merck images. The blue trace here corresponds to the intracranial field potential in response to these minimally recognizable images. These are images that are recognized, and the red ones, the red trace correspond to those that are not. And here I'm showing what happens after subjects are exposed to those full object images and when they can now recognize those submerged post images, those images that they could not recognize before. After recognition, there's a very rapid change in the brain that we can see here reflected in these intracranial field potentials. So we surveyed a large number of deep convolutional neural networks. This slide is somewhat outdated now. This was from circa 2018. You will recognize many of the top computational models in visual recognition at the time. The conclusions don't change much if you start using some of the most recent ones like EfficientNet and NASNet and so on. So the short story is these networks were able to recognize objects quite well, but they struggle significantly, even in these very simple conditions that humans excel at, which involve recognition from partial object information. So we introduced very, very simple changes into these models inspired by basic knowledge of the anatomy of the ventral visual cortex. In this particular case, we simply introduced recurrent connections at the horizontal level in the flavor of the Hopfield type attractor networks that were just presented in one of the previous talks. So here we have a deep convolutional neural network, and all we're doing is introducing within a layer horizontal connectivity. In this particular case, using symmetric weights, so the connectivity between a neuron and a neighbor neuron is reciprocal. And in this case, just by introducing this reciprocal Hopfield-style attractor network, recurrent neural network, we can improve performance significantly over existing networks without any additional training, with zero free parameters. There's no backpropping here. Okay, yes, thank you. And then if we allow ourselves to train that model, that's what we call here RNN5, we can match human performance. So we'd like to suggest that cortex, just based on these examples, might function with two modes of operation. One which is very fast, and we can think of it as a purely bottom-up response. Another one that's recurrent, takes more time, and allow us to ruminate on information and process information when you have heavy occlusion or in cases of poor visibility. So when I show this image, in addition to the fact that you can do pattern completion for each object, it's quite likely that we're also using a lot of contextual information about the image. So we're not just looking at each object in and of itself. So we've been doing a lot of work that I won't have time to do justice here on trying to understand the fact that context matters in visual object recognition and how we can use contextual cues to recognize objects. So these are just two examples of some of the failures of current computational models, how we can use inspiration from biology, from anatomy, from behavior, and from neuroscience to improve these models. And I'm particularly excited to see as outlined here in the first session, the future of computational neuroscience and the strong bridges between computational neuroscience and building biologically inspired AI. Thank you very much.
We have time for a quick question. Okay.
Great talk, thanks. How far to get to feedback? Because you showed some frontal cortex, but within the models we were talking only with vision. So where do you think the context comes from if you think about the brain?
Yeah, great question. I didn't really talk much about context. Happy to discuss further. So we have computational models of that include feedback and then try to incorporate contextual cues. We know anatomically there is feedback all over the visual system, the exception being the retina. Information doesn't go back to the retina. Everywhere else we have feedback, basically. Our contextual models are basically trying to separate processing into two streams. One basically that focuses on foveal information, the other one on the periphery. And we do include some of these feedback signals at the very least from prefrontal cortex to inferior temporal cortex, from the highest echelons. But in practice, I think, in reality, I think that we should have feedback all over, and I think that's probably relevant for visual processing.
Thank you very much.